Okay, let's talk about each different types of carbohydrates. We'll start with simple carbohydrates. Number one, simple carbohydrates, and these are going to be our monosaccharides. Monosaccharides. Okay, and this is going to be monosaccharides, and this is going to be our monomer. Okay, so if you remember the monomers, when we talk about monomers, monomers mean one, like one subunit, right? One subunit. So what makes big polysaccharides? A bunch of monomers, a bunch of monosaccharides. Monosaccharides cannot be broken down into smaller sugars. Examples of monosaccharides, like I said already, they're going to be glucose, for example. Remember that one? Glucose and fructose. Okay, the sugar that's in your fruit. Uh, Monosaccharides, like I said before, like glucose, are fuels for cellular work. In other words, they are energy. Okay, this is what glucose looks like over here. Glucose can be in a linear or a ring structure. Usually you find it as a ring structure in our bodies, all right? Usually it's in a ring structure. Um, and this is whenever you see something like this, okay, you're gonna see a ring. And remember what I told you about in a previous video. Whenever you see rings and you see these little edges here, these are gonna be carbon atoms, right? And you can actually see it here. All of these are carbons. And I know, and you will know pretty soon, that glucose is actually made, glucose has 6 carbons. Okay, so 1, 2, 3, 4, 5, and 6. There's just 6 carbons. Okay, and sometimes it's linear, sometimes it's in a ring structure, but we'll worry about this ring structure because that's usually how we see it. Now, before I move on, I want to show you something really quick as a reference. Isomers. What are isomers? Isomers are molecules that have the same formula, but they have different arrangement of their atoms. Okay, so for example, check this out: glucose versus fructose have the same molecular formula. Okay, they both are C6H12O6. Fructose is also, I'm gonna put glucose here. Fructose is also C6H12O6. So if I tell you only, if I only give you the molecular formula or the formula here, the same formula, you'll think, oh, maybe he's talking about fructose or maybe he's talking about glucose. The difference, however, comes within the arrangements. So glucose, look at glucose. What makes it different from fructose is the arrangement of this part of these atoms over here. Okay, this part here is what's different. Okay, compared to what would be fructose over here, the arrangement is going to be a little bit different. Okay, and these are actually called isomers. Again, isomers are molecules that have the same formula, but they have different arrangements. All right, let's talk about another form of simple carbohydrates, or another group, or another type of simple carbohydrates. These are going to be your disaccharides. Disaccharides. And disaccharides are okay, for two. So two monosaccharides together. So these are going to be made up of two monosaccharides. Of two monosaccharides. And how do we stick them together? Via dehydration reactions. Just by now, you should really identify these reactions really quickly, right? With dehydration reactions, that means we lose water, but we make a bond. All right. And this is lactose is going to be a disaccharide. Uh, and how do we make lactose? How is lactose made? Glucose molecules and a galactose here. This is going to be a dehydration reaction. Dehydration reaction over here where we lose water. Water is lost, but we make this bond. Okay, and that's how we make a disaccharide. So examples of disaccharides: your milk sugar, your lactose, maltose, which is in beer and malted milkshakes, and finally your sucrose, all right, which is glucose and fructose. I may not ask you what sucrose, for example, made of, but I do expect you to know that glucose, maltose, lactose are examples of disaccharides. And how do we make disaccharides, and what are disaccharides are also questions I can pose on the test. And then finally, we talk about the complex carbohydrates, and these complex carbohydrates have to do with really big carbohydrates. All right, we call them polysaccharides. Polysaccharides, which are going to be polymers, big chains, polymers of monosaccharides. Okay, in other words, long chains of sugar, long chain. Okay, in other words, long chains of sugar, long chains of sugars. And how do we make these polymers? Again, via dehydration reactions. For disaccharides, you probably just need one dehydration reaction to bring one monomer with another monomer, make it into disaccharide. But in terms of polysaccharides, you're probably gonna have to do many. Actually, not probably, you would have to go through many of these. Okay, many dehydration reactions to bring a lot of monomers together into a big chain. Um, what are examples of complex carbohydrates? Here we go, your starches. Starch, which by the way, starch is, whenever you see starch, you'll see starch is a polysaccharide. Right? Don't forget about that. It's a polysaccharide. And it's made of glucose molecules only. It's in a big chain. Starch is going to be used by plant cells to store energy. Make sure you know that. So starch is a polysaccharide. It's made of glucose molecules, and its main function is to store energy. Another example of a uh, polysaccharide is going to be glycogen. Glycogen. And glycogen, there you go. This is made also of glucose molecules. And this is going to be used by animal cells to store energy. So there may be a test question saying which polysaccharide is used by animals to store energy, or which polysaccharide is used by plants to store energy. Okay, they're different. What happens with glycogen is usually broken down to release glucose when you need energy. So, for example, you're very hungry, you haven't eaten like 10 hours, and you're hungry, you need energy, then uh, your body's gonna do something. It's gonna be a signal. It's gonna go to your liver, and your liver cells are gonna get a message saying, "Hey, break down this glycogen into glucose." So now you have glucose for your cells to use to make ATP. But for right now, just know that glycogen is another example of a polysaccharide. It's also a polysaccharide. It's made of glucose, stores energy as well, and it is broken down to release glucose when you need it. Another type of polysaccharide is going to be cellulose. And cellulose is also made of glucose, uh, most abundant organic compound on Earth. So this is the most abundant organic compound on Earth. There's why? Because there's so many plants, there's so much green out there on Earth that it's probably going to be. That's probably why there's so much more cellulose than any other thing. Um, Cellulose again is going to be a polysaccharide made of glucose, and it's gonna make kind of it's gonna be part of the cell wall, so it's gonna be very strong. All right, it's very rigid. It's gonna be a structural carbohydrate. This is something you need to know. Cellulose is not for energy storage. It's for structural. It's a structural carbohydrate. It's for structure. Okay, it's for, let's just say, it's for protection. So it has to be extremely strong. Cannot be broken down by animal enzymes. So that's why we can't really digest cellulose. We cannot really just eat plant material all the time. Some organisms, like for example, ruminants like cows and goats, and those organisms, we see them always eating grass and hay and everything. Well, the reason why is because they have microorganisms in their stomach that are going to be breaking down the cellulose. But it's not really the cow breaking them down. It's more like microorganisms in their stomach uh, breaking down the cellulose for them. Okay. However, for us, it's not like, oh, so why do we need it for? Well, it's going to be our fiber, which is great because this is going to be, it's going to give our digestive system kind of a physical workout. Okay. So for us, it's very important. For us, it's our fiber. Finally, there's another polysaccharide, which I just mentioned, which is chitin. Chitin is going to be kind of part of the external skeleton of insects or arthropods. I'm going to put here arthropods. Okay. Um, a lot of lobsters, for example, crabs, for example, insects, for example, all of those are arthropods. And they have they call it an exoskeleton or external skeleton made of this. That's why, I don't know if you ever noticed, or hopefully you never done this on purpose, but you probably done this accidentally. You're walking down the street, right? Or when there's a lot of cicadas out there, you're walking down the street, they're probably lying down there dead already, and you walk and you hear a crunch, right? Well, that's because whenever you step on an insect or something, or like cicada, for example, well, it's what you are stepping on is going to be their exoskeleton, which is made of something stronger called chitin. Okay, and also chitin can be found in the cell walls of fungi. And last but not least, I told you that a lot of these different the starch and glycogen and cellulose, and you probably you probably start thinking, wait a minute, did he say that all of them are made of glucose? So what makes them any different, right? Well, the thing is look at starch and look at glycogen. Okay, it's just about the bonds they make. You see how these bonds is just a glucose monomer, but look at the bonds here. So these bonds are kind of easy to break in your digestive system. For example, your digestive system can break them apart. Glycogen, the same thing. It's a very similar kind of bond, and also you see a little bit of branching here. Okay, so there's some branching there. Cellulose, however, it's also made of glucose, but the bonding is different. Look at how they bond with each other. All right, it's a little different. Plus, there's a lot of hydrogen bonds, which kind of also brings like different lines of these molecules together, which makes it very, very, very rigid. So it's going to be very rigid because it has a lot of bonds. So technically, if you're an enzyme, you have to break a lot of these strings. Okay, made of cellulose, and it's just going to be very hard for the enzyme to, like, for example, get in the middle of this or get in the middle of this because it's just so tight because of all of the linkages that these glucose molecules are doing with each other and with other and the other fibers or the other lines of glucose molecules.